As I had mentioned in the previous video, the urea cycle takes place exclusively in the liver, uh, so in the liver cells, the hepatocytes. And within the hepatocytes, the urea cycle is accomplished through 5 enzymatic steps. The first 2 enzymatic steps occur in the mitochondrial matrix, and the last 3 steps occur in the cytosol. So we begin here with step 1 catalyzed by carbamoyl phosphate synthetase 1 or CPS1. There is a CPS2 involved in nucleotide metabolism that I may cover in a future video. CPS1 is a pretty cool molecule because it acts like a sort of tunnel for our substrates. Our substrates feed into this tunnel, and in a uh, two-step process interacts with 2 moles of ATP. So we start here with bicarbonate ion. So in the first step, this bicarbonate ion is activated by an ATP. So it's going to conjugate to a phosphate and kick out the ADP. And the purpose of this is to make this high energy mixed phosphoanhydride. So this species is very reactive. And at this point, uh, we're going to conjugate it to our ammonia. So the ammonia is going to come in and kick out the phosphate group. So the inorganic phosphate is kicked out, and we make this carbamate intermediate. So then it's going to conjugate, this carbamate intermediate is going to conjugate to another mole of ATP to essentially make another high energy mixed phosphoanhydride, carbamoyl phosphate. So at this point, we're done with step 1, and this carbamoyl phosphate is going to feed into step 2. So step 2 is mediated by ornithine transcarbamoylase. So carbamoyl phosphate is going to react with ornithine. So ornithine is actually one of these non-canonical amino acids in that it has all the structural resemblance of a typical amino acid, an amine group, a carboxyl group, and a variable R group, but it's none of the 20 proteinogenic amino acids. So this enzyme catalyzes the conjugation of these 2 molecules. And being that carbamoyl phosphate is a high energy mixed anhydride, the inorganic phosphate is kicked out, and we get this conjugated molecule, citrulline, which is another non-canonical amino acid. So at this point, we're done with the mitochondrial reactions, and citrulline is transported out of the mitochondria and into the cytosol. So in the cytosol here, our citrulline is going to react with aspartate, an amino acid, and this is going to be mediated by enzyme 3, argininosuccinate synthetase. What's important to know here is that this enzyme is going to conjugate these molecules, and it's actually going to use ATP, but what it does with the ATP is interesting. Instead of hydrolyzing it to ADP, it actually hydrolyzes it to AMP, so adenine monophosphate, and we kick out a pyrophosphate group. So this is equivalent to using essentially 2 ATPs. And so the product of this is going to be this argininosuccinate molecule. This molecule is going to interact with step 4 here, mediated by this enzyme argininosuccinase. And this enzyme mediates a cleavage of this molecule to yield fumarate, which is actually a metabolite of the Krebs cycle, and that can feed into that, and arginine, another amino acid. Now, arginine is going to feed into the final step of this, mediated by arginase. So arginase, like argininosuccinase, is going to cleave this molecule and produce ornithine and urea. So our final product, urea, can finally be excreted in urine, and ornithine, which can actually feed back and help this cycle keep going. And so this process in total, if we were keeping count, requires 4 ATP equivalents. So 2 ATPs using carbamoyl phosphate synthetase 1, and 2 ATP using argininosuccinate synthetase.